Hi everyone, and welcome to the fourth installment on getting started using standards and reference materials in microbiome analysis. This fourth section is going to cover questions to ask when reviewing an established protocol. So moving right back into our questions that we started in the previous section, the first question we're going to tackle is, I see evidence of deficiencies in my workflow. Are they more likely arising from the wet lab side, the bioinformatics side, or both? For this type of question, we really want to use two standards. We want to utilize a whole cell and an extracted gDNA standard of the same type. And again, this is a good use for the ZymoBIOMICS Microbial Mock Community. And in this case, it's available as both a whole cell and an extracted gDNA standard. We want to use this because both of these have the same expected outputs, and both of them have a known truth. And as I mentioned, the ZymoBIOMICS Mock Microbial Community is available as both of these. And the mic score can be used equally well on outputs from both the whole cell and the gDNA standard. Pipeline-wise, we would add the whole cell mock community to our extraction process as a sample, and then we'd add the extracted gDNA standard for the mock community as part of our library preparation. Following that, when we analyze them, and we can just analyze them using the mic score as before, what I'm showing here is a triplicate for both the whole cell input and the gDNA input. So on the x-axis, we have first the triplicate from whole cell and then the triplicate from gDNA. On the y-axis, we have the mic scores generated by each individual sample. And just at a glance, we can already see the whole cell appears to have underperformed relative to the gDNA. So we can say that underperformance or that lost performance was lost somewhere in the lysis and extraction process most likely. Because when we start off with pre-extracted gDNA, we don't have the same deficiency. So that tells us that we probably need to look for some loss of performance, specifically in our wet lab technique. What if we see low performance across the board like this? Well, I'd say that I have pretty similar performance on the whole cell and the gDNA samples. Overall, they're underperforming across the board, and this is most likely lost performance that is downstream of the lysis and extraction process. Now, when I say downstream of the lysis and extraction process, on the wet lab side, that could mean some deficiency in my library prep or sequencing process, but it could also be a deficiency in the dry lab, as that comes even later, and maybe I have a weakness in my bioinformatic analysis. This particular figure couldn't tell us that, but we can start looking for that using some other methods. It's also possible I have an even more complex situation where I see underperformance across the board, but I actually see additional underperformance in the whole cell sample. In this case, we actually have some loss of performance that we can attribute to stuff that happens downstream of the whole cell standard or downstream of the lysis and extraction process. We have additional loss of performance when we start off with whole cell. So this would mean that we have deficiencies definitely in the wet lab portion when it comes to lysis and extraction. We also have deficiencies that are happening in the library prep, sequencing, or possibly bioinformatics portion, maybe even all of the above. What this will help us do is get an idea of how big the deficiencies are. So for example, if we see a much smaller step going from whole cell to gDNA, or in other words, a smaller loss of performance going between the gDNA to the whole cell, that might indicate that maybe we have a relatively low loss of performance on the wet lab lysis and extraction portion, and that we really should focus our initial efforts on the downstream portion of that. Alternatively, if we have a very noticeable step between whole cell going into gDNA, then maybe we might say we have a much larger performance deficiency in dealing with the lysis and extraction, and that needs to be our first point of interest. Another question that comes up, and this one can sometimes be one that you don't even realize you want to ask until you have a good reason to ask it. Unfortunately, often this comes a little bit too late to ask it, is I see differences in relative abundance of two microbes between samples, but how do they differ in more absolute terms? So one way that we can approach this problem, first and foremost, ensure that we're using a sampling technique that gives us the greatest possible comparability of sample input. And that would be things like having a consistent or at least a measured amount of grams of soil or feces going into the extraction process. So having a consistent amount of sample might also be, in the case of a liquid sample, maybe having a consistent volume in milliliters of wastewater or of blood or some other bodily fluid that we're measuring. This, however, becomes a lot more difficult if you're dealing with things such as a swab sample, where a swab sample can take potentially a lot or a little, especially of a drier surface such as the skin, where really you don't have a lot of control over the bacterial biomass going in. So you are potentially limited by your sample type, but obviously, to the extent possible, try and be consistent with the amount of sample going in and the way you collect it. Once we have that taken care of as much as possible, we can start adding in a spike-in standard. A spike-in standard with a known absolute quantity of microbes, and these microbes would be microbes you would absolutely not expect to see in the sample. So for this, we'd really want to select a spike-in with an appropriate range of microbial input, and by that I mean we'd want a high microbial load standard for something such as feces or soil, where we know that we're going to have a high microbial input from these sample types. For something such as tears that tend to be very low microbial biomass, we would probably want a low microbial input standard. Using our spike-in reads, we can have a bit of a yardstick to compare and maybe even get some absolute quantification too. So looking at our pipeline, we would actually add the spike-in somewhere between our sample collection and preservation point and our extraction point. So definitely before extraction happens, we would want to spike in the spike-in. The bioinformatics workflow for this is going to be very similar to the one I showed before for just generally looking at a standard. But in this case, as always, we're starting with a clean pre-processed FASTQ file. We want to make sure that those spike-in microbes are well represented in the genomic reference data, and we can assign the taxonomic values to the reads using whatever pipeline we want. Following this, we should have tax accounts, and really we want to, at this point, separate out the reads that are identified as coming from the spike-in and the reads that are identified as coming from the original biological sample. One thing that you can do that's particularly useful with the ZymoBIOMICS spike-ins is utilize the fact that these spike-ins have known inputs in known absolute amounts and multiple microbes in expected ratios. So if those ratios are off, if we know that the 2 microbes in the spike-in standard or the 3 microbes in the spike-in standard should be present in a specific ratio, and that ratio is far from expected, that most likely indicates some deficiency in the wet lab work, such as some significant bias in the extraction process. And really at that point, you should be asking yourself if you're even going to trust the absolute quantification, or if maybe there's evidence that you have such a large bias that absolute quantification is maybe not worth doing when even your relative quantifications are probably far off target. Assuming, though, that you do have satisfactory ratios coming out from your spike-in, you can then utilize the spike-in where you had a known input and treat it as a yardstick to normalize between samples and maybe even determine absolute genomic counts as appropriate or as needed. For the interpretation, as always, you want to compare relative abundances. Relative abundances are useful, but then you can do some additional comparisons by, again, measuring against this absolutely quantified yardstick that was spiked in. This is based upon a real experiment that happened. I've changed some of the major details about exactly what was being looked for and what was observed, just in order to protect the anonymity and confidentiality of the experiment that I was assisting with. But the goal of this experiment, we'll say, was to maximize the relative abundance of, or maximize the abundance of nitrogen-fixing bacteria in the soil sample type. And here we have soil 1 and soil 2 presumably after some kind of treatment. By relative abundance, it appears that we have massively increased the amount of nitrogen-fixing bacteria, having taken it from 20% relative abundance to 90% relative abundance. This would indicate success. However, if we normalize this to some spike-in that was used, such as one of the ZymoBIOMICS spike-ins, we can actually see that soil 1, again, we see the same relative abundance as before, but if we then normalize to this spike-in standard, we actually realize that whatever units we're using in normalization, we have the exact same amount of nitrogen-fixing bacteria in the soil as in soil sample 2 as 1. So the treatment did not actually increase the amount of nitrogen-fixing bacteria. What it did was just deplete everything else, and quite possibly make what I would call a more dead soil and probably a much less healthy soil. So this would indicate that whatever we tried to do was actually not successful. And then finally, one of the tougher questions to ask: can I compare my results to those of my colleagues? And this can be tricky, especially when running a multi-center trial. because obviously what you don't want to see is batch effects where different centers are always giving different results slightly and you can't compare results between centers. So first and foremost, just like I said with the absolute quantification, first thing is get your pipeline really dialed in on the wet lab side. And in the case of this, I'd say the first thing to do before even trying to measure comparability, if you have this option, is when establishing the pipeline, make sure that you're using as much as possible the same sample collection, preservation, and extraction techniques. Also match the sequencing technique and the bioinformatic analysis. That is your first and best step to make sure that you have reproducible data, have reproduced methods. Following that, I would recommend utilizing an appropriate complex reference material such as the ZymoBIOMICS fecal reference, the true matrix standard, and then determine some criteria for comparison with your colleagues. And just an example of this might be taking the 25 most abundant microbes from the fecal reference material, say all these microbes with over 1%, or all the microbes with over 1% relative abundance, and comparing between the different labs to see if they're seeing an acceptably similar result. Obviously, how you measure the result and what constitutes acceptable is up to the laboratories themselves. Selecting an analysis for doing this type of for answering this type of question. Some examples might involve clustering analyses, probably using machine learning, to identify labs with similar performance, and also to identify labs with very different performance and make sure that labs have consistent performance to themselves. Another interesting approach that's also potentially useful is anomaly detection algorithms, such as isolation forest machine learning, that will specifically identify when an anomalous sample is generated, or if a laboratory is generating samples that appear anomalous relative to samples analyzed by other laboratories. For this process, we would add a complex reference material as a sample during the extraction process. The most simple method here is just looking at firmicutes and Bacteroidetes, as well as things that are neither of those, in this complex fecal sample. And as a fecal sample, one would expect to find primarily firmicutes and Bacteroidetes. Here we can see that Lab M and Lab V appear to be just about identical. So I would say that based on what I see here, Lab M and V should be readily comparable for their data. Lab B looks extremely similar to Lab M and Lab V, so I'd say that Lab B is probably comparable, maybe with a little bit of correction. Lab Q appears to be the most divergent relative to all the others. It's still pretty similar, but I'd say if anything, Lab Q is probably the one that might need to dial in their method a little bit more, or identify what the reason is for them seeing a result that appears different than all the other laboratories and maybe address that issue. Zymo will provide a little bit of help in this area in the form of a portal for people who are using this complex fecal reference material to upload their data, and people who are starting to use this to download the previous results of others so that they compare what they see to what somebody has previously seen. And we also track documentation on things like what method was used for the sequencing and what the other properties were. So this can give you some early stage reference data if you don't have any readily available. Looking at this clustering analysis though, on the X and Y axes we just have different principal components from the analysis. And starting off, let's look at Lab 1 and 2, and we see Lab 1 and 2 clustering very nicely down in that lower right corner, and in fact their data basically overlapped. I couldn't really draw a shape that would differentiate between the two labs, at least with the 3 points I have for each. So I'd say Lab 1 and 2 should very reliably be able to compare results with each other and get meaningful comparisons. More likely than not, they've worked pretty hard to ensure some consistency between the two of them. Labs 3 and 4, they cluster together very well. In this case, I would say the laboratories are distinguishable. I could draw squares that would divide Lab 3 from Lab 4, but they are still clustering close to each other. Also very important to note, while Lab 3 and 4 look like they're probably comparable, or at least pretty comparable to one another, they wouldn't be as comparable as Lab 1 and 2, and they're also clustered quite far away from Labs 1 and 2, meaning that I would definitely not want to compare data from Lab 1 or Lab 2 with Lab 3 or Lab 4. Finally, moving on to Lab 5, this one is kind of our problem child. And what we see here is, number one, a much wider cluster for this one laboratory than any other laboratory shown here. It also clusters apart from any other laboratory. So I would say that Laboratory 5 is generating data that's not comparable with any other lab, and in fact is also not necessarily internally consistent within the lab itself, as we see a wider dispersion of the data points than what we see for most other laboratories. So I would say Lab 5 probably has some deficiencies in its method and may need to address that. Now it's time to talk about a deeper dive into the bioinformatic approaches. But for that, we're going to have to wait until the next session. So I'll see you in session 5 with a discussion of how we approach the bioinformatics of these methods.